everyone, I'm Ruba Bayram, a first-year medical student and a member of PACademia. Today, I will be discussing about the immunotolerance chapter. I hope by the end of the video you guys find it helpful and will clear some of your doubts. So the first thing I want to talk about is tolerance. What this tolerance means? Tolerance means the unresponsiveness to an antigen. So my immune cells are not going to be responding to a certain antigen. They're not going to be recognizing this antigen. What type of antigens do I want my immune cells not to recognize? Three things. I want tolerance against my self-antigens. So my immune cells are not going to be recognizing my self-antigens. They're not going to be reacting to them. That is good. I don't want this to happen because if they do, if my immune cells recognize my self-antigens, then they will start to attack them. This is what happens in autoimmune diseases, okay? So my immune cells need to be tolerant to my self-antigen. Unresponsive, do not recognize my self-antigens. Secondly, my immune cells need to be tolerant towards my harmless environmental antigens, which are the commensal microbes. Commensal microbes. These are going to be the normal flora that is found in the GIT. They're very helpful. I don't want my immune cells to attack them and get rid of them, okay? A third thing is to avoid excessive lymphocyte activation. This is when I am going to be, see, here says normal protective responses. This is during normal protective responses. Let's say I am infected with a pathogen. There needs to be a limit on how much lymphocytes are being activated. Let's say it's been 2 to 3 days. I got rid of all the pathogens. There's only pathogen remains. What is this? Let's say I have this human is the pathogen. This is an example. This human is a pathogen. My lymphocytes will start to break it down. It will break it into a head, a leg, arm. Are these useful by themselves? No. Okay, so they're just remains. They're not going to be doing any of that. These pathogen remains are not actually dangerous, you could say. However, APCs or dendritic cells they will just catch any antigens they find. Self-antigen, actual pathogens, pathogen remains, they will collect anything, any antigen they see. If the APC presents a pathogen remains, I don't want my T cells to be activated more because I already destroyed this pathogen. It's just remains. Okay, so this is when I need my tolerance to happen and regulation to happen. Okay, obviously I don't want excessive lymphocyte activation in these conditions because then it will cause over-inflammation and this can cause what? This can cause tissue damage. Okay, three types of antigens tolerance need to be against: self-antigen, my commensal microbes, which is the normal flora, and then to avoid excessive lymphocyte activation, which is in this case pathogen remains. Again, definition: unresponsiveness to an antigen, as I mentioned. Significance: self-tolerance, and it's pretty self-explanatory. Okay. Now, what I need to explain is the tolerance happens in two different places. There's something called central tolerance and peripheral tolerance. Central and peripheral, let's try to understand what that is. Central is going to be the generative lymphoid organs. From the name generative, so these are the organs that generate these lymphocytes, which are the primary lymphoid organs, aren't they? This is the thymus and the bone marrow, okay? The thymus and the bone marrow. This is the central. In the peripheral, it is the rest, which is the what? It is the secondary lymphoid organs. Okay, which is the spleen, the malt, the lymph node, all of this. So the tolerance happens in two different places. I have it happening in the central and the peripheral. Okay, I have made a table that is going to be pretty simple to understand. So I'm going to be explaining all that. As I said, there's central tolerance and peripheral tolerance. Tolerance that is happening in the central lymphoid organs, which is the primary lymphoid organs, the thymus and the bone marrow, and the peripheral tolerance is in the peripheral lymphoid organs or secondary lymphoid organs. Central tolerance is going to be of the T cell and B cells. As I mentioned, the immune cells are not supposed to react to the antigen, the self-antigen, okay, self-antigen. The immune cells are not supposed to recognize or respond to the self-antigen. So if they do, if the T cell, now I'm talking about the T cell, if the T cell strongly recognizes the antigen, so I have the self-antigen, okay, self-antigen, I'm talking about self-antigen. I want tolerance against my self-antigen, yes? So I'm talking about self-antigen, and this is my T cell. If the T cell recognizing self-antigen very strongly, it will undergo apoptosis death. Make sense? I don't want my T cell to recognize the self-antigen. I don't want it to recognize it because it can cause autoimmune disease. So if it strongly recognizes, right away I'm killing this T cell. I'm getting rid of it, apoptosis, okay? If it is a weak or slight recognition, or basically the T cell will recognize the self-antigen just a little bit, just slightly, development of regulatory T cells happens. So you either strongly recognize the T cell, the self-antigen, sorry. The T cell either recognizes the self-antigen strongly or weakly or not at all. Not at all, nothing will happen. It will be positively selected. It will survive. But if there is any type of reactivity towards the self-antigen, this is where something is happening. If it is very strong reaction to the self-antigen, apoptosis happen, death. If it is weak recognition, then regulatory T cells are going to be formed or developed. In the case of B cells, it's the same thing. It either strongly recognizes the self-antigen or weakly or not at all. If not at all, normal survival, positive selection. So there's only two options: strongly recognizing the self-antigen or weakly. If the B cell is strongly recognizing the self-antigen, you would expect, okay, apoptosis. But in the B cell, there is a chance that is given. The B cell is given a chance to do some editing. This is known as receptor editing. This is where the receptors of the B cells are going to be edited and changed. If after this editing, the B cell still recognizes the self-antigen, then apoptosis happens. So the outcome is still the same, but the difference is B cell is given a chance, T cell is not. Okay? And if there is weak recognition, anergy happens. If the B cell recognizes the self-antigen weakly, anergy happens. So you learned this table pretty easy. T cell strongly recognizing self-antigen, death, apoptosis. Weakly, T regulatory cells have development. B cells, if strongly reacting to the self-antigen, then what? Then it is going to be a chance is given to them, receptor editing happens, and then apoptosis after that. If after this chance that is given to them, they still recognize the self-antigen. If it is weak recognizing, the B cell is recognizing the self-antigen weakly, anergy happens. In central tolerance, keep this in mind. Now, if I ask you a question, anergy happens in central tolerance of which type of cells, T or B cells? You need to confidently tell me B cells. Anergy in central tolerance. T cells, central tolerance, what happens? Either apoptosis or Treg development. B cells, either apoptosis, obviously after undergoing the second chance of receptor editing, or anergy. So here you need to know. Peripheral tolerance will be the same in both cells. What happens in peripheral tolerance? Three things happen. Either anergy happens, either suppression happens, or either deletion happens in both T cells and B cells. So the only difference we see is actually in the central tolerance, okay? In the central tolerance. Now, in anergy, what we can notice is here, there's going to be no T regulatory cells are found. The suppression and anergy, so deletion is pretty obvious. If there's strong reactivity to the self-antigen, apoptosis, you get rid of it death. But I'm talking about the anergy and suppression. What is the difference? The anergy and suppression, in the anergy, we are only making use of the T cell itself, or the B cell, whatever, but the cell itself. It doesn't require any help from external sources. In the suppression, we are making use of the regulatory T cell. So the only difference is the environment. If a Treg is being used, it's a suppression. If an anergy, if there's no Treg, then anergy happens. Let's look at each case. Now, T cell anergy. If I tell you T cell anergy, where is this found? Peripheral or central? Peripheral. Okay, central of T cells. It's only what? It's only the apoptosis or the Treg development. Okay, T cell energy is in peripheral tolerance. Okay, what happens? I told you energy is going to be only the use of the cell itself. It doesn't require help from Treg. So we can see that here. APCs, dendritic cells, will collect antigens, self-antigens, pathogens, pathogen remainants. You remember the remainants I told you about? The head just leaving, the arm, the just remainants. If the APC or the dendritic cell is holding or presenting a self-antigen, the T cell is going to be, there's a signaling block that happens and the T cell will undergo energy. Are we using Treg? No, this is energy. No Treg is used. Treg is only used in suppression. Tamam? So now you tell me, how did you know this is a self-antigen? Because there is no B7. B7 receptor is not found. And as we know, B7 receptors are only found when there is going to be a pathogen, not when there's going to be a self-antigen. If self-antigen is presented, no B7 is there. So here, because there is no B7 is not here, then the T cell will notice and is going to be what? Unresponsive. There's a signal. The second way is by using inhibitory receptors. How is this used and when? Here, this is when the APC or the dendritic cells, which is this, is expressing or showing what? Pathogen remainants, okay? I told you that we need tolerance against the remainants of the pathogens, right? And there's three ways: self-antigen, or the normal flora, or the pathogen remainants. This is where this is happening. The APC, the dendritic cell, will just collect any antigen. It doesn't know. So it might collect just a pathogen remainant, just a leg. Example is a pathogen doesn't actually have a leg, but in this case, let's say, catches a hand. It's already dead. It's just remainant, but it's still going to show to the T cell. The T cell will know. It's going to say, okay, I have already killed this pathogen. I need to be what's not activated. I need to be inhibited. And that's where we use the inhibitory receptors. Okay? How did you know? You tell me, how did you know this is a pathogen remainant? Easy, from the B7. B7 is expressed. Because B7 is expressed, this means it's not a self-antigen, obviously. So it is a pathogen remainant. Both ways, energy will happen. In both ways, either using the signaling mechanism or I'm using inhibitory receptors, both of them will end up in energy, which is the T cell will be unresponsive. This is the explanation of the whole thing. Here you can see that it says, if the T cell recognizes a self-antigen without co-stimulation. What is the without co-stimulation? This. I'm talking about the B7. If there's without co-stimulation, if there's no B7, CD8 signaling, the T cell will identify, will be able to understand, okay, there's no co-stimulation. Because there's no B7, this means I have to be unresponsive to this. So it will have the signaling blockage. So a block of signaling. Or if it was a pathogen remainant that is being presented, then it will use inhibitory receptors. The ones that we will talk about is the CTLA4 and the PD1. Specifically, though, CTLA4 is what we're looking at. So we're done with energy. Energy is not using the Treg, it's only by itself. Two ways, signaling block or the use of inhibitory receptors. Suppression. I told you guys suppression is the involvement of the Treg, the regulatory T cells. Regulatory T cells will suppress. They will suppress in two different ways. That's why there's two different arrows you can see here, meaning there's two different pathways. Either it releases cytokines or uses inhibitory receptors as well. Okay? Now let's talk about regulatory T cells. As I mentioned, I told you guys regulatory T cells are formed when they're formed when they are going to be recognizing the self-antigen weakly. So recognition of self-antigen, weakly. The self-antigen recognition weakly can happen in what? Two different places, either in thymus or in the lymph nodes. If the T cell, and I'm talking about CD4 specifically, by the way, if the T cell is going to be recognizing weakly the self-antigen in the thymus, it will be known as the natural Treg, N-Treg. If it is going to be recognizing, if the CD4 T cell is going to be recognizing the self-antigen weakly in the lymph node or any primary, peripheral lymph node organ, then it will be known as the induced Treg, Treg. This is the whole concept. T regulatory cells are formed in two different places, either the thymus or the lymph node. In the thymus is not natural Treg, and in the lymph node is the, or any peripheral lymph node organs, it is the induced Treg, Treg. Now here, this idea is that the regulatory T cell, I will keep referring to this as Treg because it's easier. The Treg will have markers that differentiate it from other T cells. How will I know this is a T cell, T helper cell, this is a CD4, CD8, this is a Treg? How will I know? From markers. This is a gene that is found in the Treg, FOXP3. That's it. This is the idea. As I mentioned, the Treg, what is the function? Suppression. What is it suppressing? It's either suppressing or inhibiting the T cell from being activated. Okay. If, let's say, the T cell was activated, then the Treg can suppress the already activated T cell to prevent it from, being activated even more. And it can even inhibit or suppress the natural killer cells, the B cells, and more, like macrophages, dendritic cells, and all of it. This is the same idea. So how are they generated? In two different places. If it's in the thymus, it's a natural regulatory cell. If it's in a peripheral lymphoid organ, it's a inducible regulatory cells. What are the function? Again, suppression, inhibition, all of this. Okay. Now, inhibitory receptors of the immune system. I mentioned that the immune system is going to be inhibiting. It can be inhibited by inhibitory receptors. Okay. What are these inhibitory receptors? We mentioned the CTLA4 or the PD-1. Okay, the CTLA4 or the PD-1. In this case, this is not really important, but the idea here is that cancer cells, what they do is they start to form these inhibitory receptors so that the immune cells are not able to recognize them. When the cancer cells have a lot of inhibitory receptors, the immune cells will be inhibited. So they will no longer be able to kill the cancer cells. So these scientists found that out, and they started to make these drugs that will downregulate these inhibitory receptors. So what these drugs do is they just get rid of this inhibitory receptor. They prevent this cancer cell from making this inhibitory receptor. And because there is no longer this inhibitory receptor, the immune system is now activated. And now it can kill the cancer and get rid of it. Okay? Now, as I mentioned, there is the CTLA4 and PD-1. CTLA4 will be expressed, make sure you know this, on both T cells, and I'm talking about activated T cells as well as on Treg. CTLA4 will be expressed on these two. I already talked about how Treg will be suppressing in two different ways, the cytokines and the inhibitory receptors. This is one of them. Now, in the activated T cells, will be telling me, okay, where is this? Where did we talk about this? We talked about it in the T cell energy. You remember I told you guys we're going to be incorporating using inhibitory receptors. This is it. Okay, this is it. All right, so the CTLA4 will be expressed on two different cells, the activated T cells, as well as the Treg. So keep that in mind. The PD-1 will have the same idea. It will be obviously expressed on T cells, and then it will do the same idea. It will inhibit. Okay, but our main focus is CTLA4. Now, how does this function start? Now, as we know, if there's a B7-CD28 interaction, then this means this is a co-stimulation. This is going to be activating the naive T cell to proliferate and differentiate. This is activation. But I want inhibition. CTLA4 is an inhibitory receptor. What happens? How does this inhibitory receptor, how does it inhibit the immune cells or the activation of the T cells? So what happens is that CTLA4 is going to be expressed, and CTLA4 will start to compete with CD28. CTLA4 will always win. Okay, so I have CTLA4 as well as the CD28, and they're competing against what? They're competing for who? For the B7. They're competing for the B7. Who's winning? CTLA4. CTLA4 always wins. Why? Because it has higher affinity to the B7. Okay? Okay. So as I told you guys, okay, this is normal. This is when there's activation. But let's say the CTLA4 is being expressed. When the CTLA4 is expressed, it will compete with the CD28, okay, the CD28, this is CD28, it will compete, this CTLA4 will compete with the CD28 for the B7. And always CTLA4 will win because it has higher affinity to the B7 when compared to the CD28. طيب, what does it do? CTLA4 is an endocytotic receptor. This means that it will endocytose, meaning that it will take and pull this B7, it will engulf it inside and it will start to digest it. As you can see here, it started to engulf it in a vesicle and it will start to digest it. Keep in mind, look how this is reminded every second. T cell, the CTLA4 is expressed on activated T cell or Treg. CTLA4 is found on these two cells, so this is repeated multiple times. Now, you might be asking, why am I getting rid of the B7? I could just, when there's competition and CTLA4 is always winning, this means CD28 will never be able to bind to B7 for activation. So خلاص, I won. Why am I getting rid of the B7? The reason is because we want the APC to not be able to activate other T cells. This is the reason. So it is a great and very smart function because it is going to be preventing this same APC from activating other T cells. Because I'm getting rid of the B7, how is it going to be activating any other T cell? So this is a smart mechanism. It's not only suppressing or inhibiting this specific T cell, but it is preventing that APC from activating any other T cell. How? By removing this B7. Okay? Again, it's an endocyte. CTLA4 is an endocytotic receptor, meaning what? It will remove the B7. Obviously, it will bind to it first, and then it will remove and ingest it. As I mentioned, the CTLA4 will be expressed on either the Treg or activated T cells. I mentioned this many times, and it will outcompete CD28. So it will compete with the CD28, and it will always win. And why does it win? Because it has a higher affinity. CTLA4 has a higher affinity to the B7. That's why it always wins. Now here, this is very self-explanatory as well. Very simple. There's cell intrinsic and cell extrinsic. What does this mean? Cell intrinsic is the idea that the CTLA4 is expressed on what? The activated T cells. Whereas if the CTLA4 is expressed on the Tregs, it's known as the cell extrinsic. Can you see? Treg. Okay? You can see regulatory T cells. Whereas this one, this T cell, activated T cell is expressing the CTLA4 by itself. It does not require the Treg. So this means this is what? Energy. We remember, and this is suppression. Why is this suppression? Because we are using regulatory T cells. Okay? This is just explaining the whole thing I explained. So two different cell intrinsic action of CTLA4 or cell extrinsic. Intrinsic, meaning the CTLA4 is expressed on the T cell, the activated T cells. Whereas the cell extrinsic is when the CTLA4 is expressed on the Treg. Okay? Functions of the CTLA4, it obviously limits activation, all of this. Okay? Now, how does the T cell know when to express CD28 or when to express the CTLA4, which one to express more? This is with the help of the B7. Pretty self-explanatory. If I have a high B7, I told you guys this means what? A high B7 is when I have a pathogen. When the APC is, this is my dendritic cell. Okay, dendritic cell, easy. Okay, it has the MHC, and here is my T cell. It has a TCR. This is the first signal. If the B7 is present, this is the CD28, okay, then obviously this will be happening. When is B7 expressed? When there is a pathogen here. But if there's no B7 or a low amount of B7, I told you this means that the DC is what it is going to be expressing or showing self-antigen. And if it's a self-antigen, no B7 is going to be happening, or just very low amount. So if it is a self-antigen, I want to inhibit it, right? I want to inhibit. So that's why I express CTLA4 more. Again, peripheral tolerance in B cells, I have explained it, is the same thing as the T cell. We have the energy, we have the deletion and the suppression using the inhibitory receptors. Okay, I hope you guys have enjoyed the video and understood the concept. Thank you guys for listening.